All right, let's dive in. Today, we're going to decode the blueprint of life itself. We're going to unlock some of the amazing secrets hidden deep inside our DNA by exploring the powerful world of molecular markers. Think of them as tiny signposts that can tell our entire biological story. So, have you ever stopped and wondered how a tiny drop of blood at a crime scene, or a simple cheek swab for an ancestry test, can reveal so much? I mean, how can a single cell tell a whole story? Well, that's the big question we're tackling today. You see, the secret isn't reading all 3 billion letters of the human genome every single time. That would be insane. It's all about knowing where to look. Imagine the genome is this massive encyclopedia. If you wanted one specific fact, you wouldn't read it cover to cover, right? No, you'd use the index. It's all about finding the right bookmarks, those specific tags that point us exactly where we need to go. Okay, so what are these bookmarks really? That brings us to our first section, where we're going to get right to the core of it. What exactly is a genetic marker? To put it simply, a genetic marker is just a known location on a chromosome. It's a tag that lets scientists pinpoint a precise spot. They call it a locus in the massive, sprawling landscape of the genome. It's kind of like having a unique street address on a road that stretches for millions and millions of miles. But, a street address isn't very useful if every single house on the street is identical. And that's where this next concept comes in, and it's absolutely crucial: polymorphism. Poly for many, morph for form. It just means that at this specific genetic address, there are different versions, or what we call alleles. This variation is what makes us, well, us. It's what makes each of us unique. And this leads to what is probably the most important rule in this entire field. Without that variation, without polymorphism, these markers are completely and utterly useless. I mean, think about it. If every single person had the exact same DNA sequence at a marker, it couldn't tell us anything about what makes one person different from another. So the key takeaway here is variation is everything. Now, the way we find these markers has evolved so dramatically over the years. We've gone from just, looking at an organism's traits to being able to read its DNA sequence directly. So let's take a look at how the scientific toolkit has changed over time. This slide really tells the whole story of that evolution. We started way back with morphological markers. These are things you can physically see, like the color of a flower. Then we moved on to biochemical markers, which were a bit more advanced, looking at things like proteins. But the real revolution, the real game changer, came with molecular markers, which finally let us read the DNA code itself. So you might be asking, why the big shift? Well, those older methods, they had some major drawbacks. For one, they were often really influenced by the environment. Was a plant struggling because of its genes, or did it just not get enough sun? Hard to tell. Plus, a lot of the time, there just wasn't enough variation to be all that useful. We needed a tool that was way more direct and reliable. And that brings us to the really cool stuff, the molecular toolbox. Let's pop the hood and take a look at a couple of the key techniques that literally changed everything. Okay, our first example is a classic one called RFLP. You can just think of it as the molecular scissors approach. Now, the full name is restriction fragment length polymorphism, which is a total mouthful, so don't even worry about it. Just remember molecular scissors. And check this out. This slide illustrates perfectly how it works. We use these special proteins called restriction enzymes, our molecular scissors, and they only cut DNA at very specific sequences. So look at the top diagram, chromosome A. The scissors make 2 cuts, which gives us 3 fragments. But now look at chromosome B. A tiny little change in the DNA code got rid of one of those cut sites, so now the scissors only cut once, leaving 2 bigger fragments. And those different fragment sizes, that's the polymorphism we can actually see and measure. So in the lab, the process follows a pretty clear sequence. First, you digest the DNA with those enzyme scissors. Then, you separate all those fragments by size using a technique called southern blotting. After that, you add a labeled probe, which acts kind of like a magnet, sticking only to the gene you're interested in. And finally, you can visualize the results and actually see those different fragment sizes. All right, let's move on to our next technique, and this one is honestly even more powerful. It's called simple sequence repeats, or SSRs. I love this one. I like to call it the genetic stutter because it's looking for parts of the DNA where a short sequence is just repeated over and over and over again. The polymorphism here is just so elegant and simple. It's just the number of repeats. That's it. Take a look at genotype A versus genotype B. Maybe genotype A has a sequence like ATATAT that's repeated 18 times. But genotype B has that same sequence repeated 22 times. That simple difference in the length of the stutter creates an incredibly accurate marker. And this is what it actually looks like in the lab. How cool is that? See that image on the left? Each of those vertical lanes is a different person. And the dark bands, those are the markers showing the different repeat lengths. Just look at how much variation there is between individuals. These unique little ladders create a distinct genetic fingerprint. And that's why this technique is so powerful for identification. So we have these amazing, powerful tools. But what's the point? What do we do with them? Let's circle back now to the big picture and see how these markers are actually put into action. Why does any of this really matter? Well, the most famous application is probably genetic fingerprinting, which we use to answer all sorts of questions. How diverse is a population? How are these two individuals related? And is that Honeycrisp apple you bought at the store actually a Honeycrisp? Yep, markers help us verify all of it. And the impact in agriculture is just as massive, all thanks to something called marker-assisted selection. See, the old way meant waiting years for a plant to grow up just to see if it had a trait you wanted, like drought resistance. The new way, you just test the DNA of a tiny seedling. This completely revolutionizes breeding. It allows scientists to create better crops way faster by stacking or pyramiding multiple good genes into one single super variety. So when you step back and look at it all, from solving crimes to feeding the planet, molecular markers have given us this incredible ability to read the blueprint of life with stunning precision. And that leaves us with one final, pretty fascinating question to think about. Now that we've learned how to read the blueprint, what should we build next?